This is the presentation that I am giving today about the molecules that shape nuclear medicine at the 21st Biannual Congress in South Africa. I am really happy to be home and to talk about this topic. It is about the past, present, and future of nuclear medicine, which is quite a big presentation. So that's why I also record it now so that it's available later, because it is possibly a bit of an unstructured little bit of everything that's my favorite in nuclear medicine from start to finish. And yeah, I have an hour and a half to talk, but this is a lot of information and perhaps better also to look at it a little bit later and revise afterwards. So yes, of course, I'm affiliated with the Catholic University of Leuven at the moment. And yeah, it's in 2025, so maybe later when you watch this presentation, it has changed already a little bit because nuclear medicine moves really quickly, as we all have seen in the last few years. So let's start with the past, where did nuclear medicine all start, and it's important to also remember the people that shaped nuclear medicine. For example, Henry Becquerel that discovered natural radioactivity for the first time. That's why he's also honored with a standard unit of his own. And then Marie Curie was inspired to investigate radioactive elements further and discovered many more isotopes, for instance, polonium and radium. And then building on their work was George Davison, that was the first pioneer to think about the use of radioisotopes as radio tracers. So we owe it to George Davison that we sometimes talk about radio tracers. But of course, there was no understanding of the risks. I always laugh at this, like radioactive thorium, which, as we know, is quite a dangerous alpha emitter, was viewed as perpetual sunshine and was included in energy drinks. Maybe these days this is more what I need because coffee doesn't seem to be doing it anymore. We had radioactive cosmetics. And as you can see on the tin, it was supposedly supported by Alfred Curie, but he was actually not supporting this and many times had some case against the people that used his name to sell the product. But as you can see, he was quite a popular figure in 1932 because he was well known. And then also, and that's why it was also used as a gimmick for advertisement. And there was also the radioactive toothpaste that was killing possibly the bacteria, but many more other things quite efficiently, also containing some thorium. Luckily, now we know the measures of radiation protection is time, distance, shielding, and the amount of dose. So whenever you want to save yourself from radioactivity a little bit, you spend a lot less time near sources. This can ensure that you maybe get less exposed. So what this means is that when you do work with radioactivity or radioactive patients, you do your work as quickly as possible, safely and efficiently, not spend a lot of time hanging around in radioactive areas without working with the radioactivity currently, and also practicing the procedures, what you will do cold, so that you get efficient on handling radioactivity before you start, because it has been really shown that the more experienced you are, the better and more quicker you do your work and the less exposure you get. Distance always means to make sure that you put as much distance as possible from you and the source. And we're also, of course, talking about the inverse law, where you get a quarter of the exposure less by halving the distance, doubling the distance, sorry. And then working with tweezers, just spending again less time close to sources, and also just designing your facility in a way where, say, the very high radioactive areas like injection rooms and so on are quite far away from other areas. Shielding is very important, but also the right shielding for the right isotopes that you're working with, like the difference between beta minus emitters shielding and also um, shielding for gammas with lead, and just maybe alpha emitters not really needing shielding, but shielding is always very important and sometimes often neglected, and this can do a lot to protect the safety of your operators. And then also the dose. I always add the dose and say you should work only with the activity that you need. This is not always part of the normal textbook ways to protect yourself, but say you elute the gallium generator and you will only label for one patient, but you have enough gallium for 3 patients. For me, it's best to remove the gallium you will not be using for labeling and put it already in the waste that is shielded rather than labeling for 3 patients and then only using one. Of course, we came to the agreement of those limits. For me, the dose limits is quite a controversial or difficult concept because in some instances, people can think that we do not need to worry if my dose limit is 20 millisieverts and I get 90 millisieverts a year, but this is in fact not the reason for dose limits. We always need to be as low as reasonably achievable. So if you can work in a way where you only get 5 millisieverts a year, there is no reason to work in a way where you get 19 millisieverts a year. So you should always focus on radiation protection wherever you are, wherever you work. It should be a culture of safety. But just to say that should you reach more than these limits, you are no longer allowed to work. So this is per year. 20 millisieverts for the whole body, 20 millisieverts for the lens of the eye, because we are worried about um, deterministic effects and cataract formation, and in the skin, as well as hands, forearms, and feet and ankles. And then we start with the history part where I explained to you the first use of radioactivity ever was with the beta minus emitter phosphorus 32. So of course, George Davis, he was working on biological tracing of biological systems with phosphorus 32. It is a beta minus emitter with a 66-hour half-life and a 1.71 millisievert, milli-electron volts decay, which is quite a strong, on the stronger side of beta minus emitters. And interestingly enough, phosphorus naturally gets targeted to bone formation, but also when it's injected intravenously, it can not specifically target malignancies of blood origin. Of course, it has bone marrow suppression because of the targeting to the bone and the rather in-depth penetration of beta minus emitter. And so these days it's no longer popular to use phosphorus 32. Also, that is a picture of the first cyclotron that was used to make this phosphorus 32. And this was done in 1946, and just to explain then also the differences between beta minus emitters and alpha emitters. Alpha emitters is a heavier particle that causes double-strand DNA breakage, has a shorter range, but also a higher energy deposit per range, and it's less sensitive to hypoxia because it relies more on just double-stranded DNA breakage rather than the formation of reactive oxidative species. So it's better for tumors that are homogeneous. Because of the shorter range, there has to be receptor expression throughout the tumor. So the alpha emitters is deposited everywhere on this tumor, whereas beta minus emitters maybe do not need this most homogeneous expression, but rather the longer range will affect cells close to the tumor where it got targeted to the receptor, as well as maybe even as far as 10 millimeters. But it causes single DNA breakage, and if the tumor is more resistant, this DNA breakage can sometimes be repaired. Um, but we are also looking at other effects like the activation of the immune system, bystander effect, and all of these things. So there's a lot to consider choosing between beta minus emitters and alpha emitters. And it depends on the clinical picture, what is the choice. And also, of course, I think even more is the availability of the radionuclides you want to use. So just to give you some information about beta minus emitters, and there is many that has been used over the years. This table, I think during the actual presentation today, I will not discuss in depth because it's a lot of information. But at the moment, lutetium-177 with a 6.7 half-life and the Betamax of 0.5 here is the one that is mostly used, the most available and most studied. It has a range of 1.8 millimeters, and this makes it having less side effects of bone marrow suppression and kidney toxicity because the range is shorter than most beta minus emitters. So if you look at this, of course, you will see here terbium-161 has very similar characteristics, shorter range though. So it can be even milder on the bone marrow and so on, and maybe this can lead you to be able to administer more radioactivity for a higher dose to the tumors. So in all other instances, even in the chemistry, lutetium-177 and terbium-161 is quite equal. Samarium-153 has been used a long time for the targeting of bone pain palliation. And then rhenium-188 comes out of a generator. It's one of my favorite isotopes, but really not widely clinically applied. Yttrium-90 is applied from time to time. And then there's the strontium-89 and phosphorus-32 used also for bone pain palliation, but you can see the beta max range of both of these isotopes or radionuclides is quite big, quite far, and this can lead to additional toxicity and side effects. Iodine-131 was then first used in 1941 and for therapy in 1946, both for imaging and diagnosis. And normally you will hear that iodine-131 was the first true theranostic application. Theranostics being where you use something to diagnose, so you image the disease, and then with the same type of molecule or targeting mechanism you treat. This is physiological targeting, so it wasn't actively targeted, and so we can discuss for a very long time if this is targeted therapy, but iodine-131 has still application very widely today and is the most commonly used therapeutic radionuclide. It was later realized that, of course, based on organic chemistry, well-known chemistry of iodine, that we could, through electrophilic substitution, label iodine-based molecules, of which the most well-known one is MIBG. And sodium iodide is then used to exchange the hydroxy group and actually do a whole exchange interaction for MIBG. And just to follow up on that, it's important to know that the free iodine is taken up through the sodium iodide symporter, and this then leads to a natural accumulation in the salivary glands, thyroid glands, stomach, intestines, and bladder. And so unstable molecules can be quite toxic. The first targeting then was MIBG, as we discussed, and then you can see the change in biodistribution when MIBG is now labeled with the iodine-131, is targeted through MIBG in the norepinephrine transporters. There's also many other ways like passive diffusion and using the VMAT2 system. And so there is uptake in the sublingual glands, the brown fat uptake, also the gastrointestinal system, still a little bit, the heart, excretion in the bladder, and of course the liver. It is used for a lot of applications, including neuroblastoma. The first more advanced targeting was done through monoclonal antibodies, and this is like immune reactive molecules that are produced that target single receptors. It is the most specific way to target radioactivity, even being more specific than peptides and all of the other molecules being developed. Um, and you normally have a linker and a chelator that then ensures that your radionuclide is coupled to this biological molecule without changing its pharmacokinetics, and this can then target any disease, for instance cancer, for which there is a immune reactive response in the body. Yttrium-90 is a pure beta minus emitter with a 64 hours half-life, which matches the pharmacokinetics of monoclonal antibodies and a 2.3 MeV emission. So it's ideal for deep tissue penetrations. And mostly Zevalin was used for non-Hodgkin's lymphoma. Suddenly, everything changed with the application or the making availability of technetium-99 as the first diagnostic, and also the development of the Anger gamma camera to image. And this is where the diagnostic uses of radioactivity first gained momentum and shaped what we have today in nuclear medicine. Just to give you an idea how the different imaging technologies caught up. So in 1940 to 50, the Anger camera was developed, which is also in the planar cameras. And then thereafter, PET was developed in 1950s to 60s, but it took a very long time to implement because the cyclotron technology was not up to standard to produce the isotopes you need for positron emission tomography. It is, of course, three-dimensional and functional. And later on in the 2000s, the hybrid cameras came that also gave you the anatomical information by incorporating CT and even later MRI. SPECT started in the 1970s with the rotation devices that could give you also 3D images, and CT came also even later in the 1990s, which of course today SPECT CT has quite an important role to fulfill in the symmetry calculations of the beta minus emitters like lutetium-177. PET MRI is just very expensive and used mostly for research purposes, more often for things like neuroimaging and situations that really requires more information and soft tissue um, imaging. Okay, so just to recap, what is single photon emission tomography? It is the emission of one single photon that is released from the area where it was accumulated, but it doesn't give you really accurate depiction of where exactly in the body this emission came from. And so you have these filters that are big lead filters with holes in, and if the ray hits this filter parallel, it will go through. If it's not parallel and it hits the filter in any other angle, it will not be registered. What this means is that the resolution of SPECT is always a bit lower than other imaging possibilities, because you will lose a lot of emissions that is just not registered and calculated for imaging. At the same time, the technetium generator contributed to the use because, of course, you need also the radionuclide, and it's a very convenient way to have a source of radioactivity on site that you can elute from time to time and just get activity and label. So the chemistry of technetium was very useful, and the radionuclide became widely available. Although we sometimes have some shortages in technetium, which is critical, 80% of nuclear medicine procedures done in the world still uses technetium, and it will never disappear, of course. So you have a system that is a column that works by chromatography, and there is a change from the parent isotope's chemistry to the daughter isotope of technetium. So the molybdenum is just is loaded onto the column and will stay there until it decays to the daughter technetium, where it's then no longer having the affinity to the column, and it gets released from the column and can be washed off by sodium chloride. It's always covered in thick lead shielding for safety of the operator and filtered for sterility, and it's nearly the ideal generator for the use in the clinic. Very easy to use, very safe for the patient, and gives a physiologically acceptable product that you can inject into the patient. And on this right side, you will see the normal biodistribution of technetium, with the thyroid being illuminated, also the glands, um, the thyroid, parathyroid, and also salivary glands, and then you also see gut uptake. Very importantly, to do quality control on your ileus. This will be discussed. I actually have a presentation on technetium on my YouTube channel that you can have a look if you want to have more in-depth information. But basically, we monitor the difference with, we use the differences in emission, with molybdenum 99 giving higher energy beta minus emissions of 740 and 780 keV. So we shield 140 keV emissions of the pertechnetate and measure just what is left, and then we can subtract, and we can calculate the difference between molybdenum and technetium 99. Very importantly, is this is not a static number, because molybdenum 99 decay by 66 hours half-life, whereas technetium 6 hours. So this relationship between technetium and molybdenum is different at different time points after elution. So it's always important to correctly calculate at what time it will no longer adhere to the specified criteria of the United States pharmacopoeia of 0.15% or the European pharmacopoeia of 0.1% of molybdenum presence in technetium 99. So, how do we label? A technetium kit normally has a ligand, a reducing agent, a buffering agent to get to the correct acidity level, antioxidants and stabilizers for stability purposes, and then a lyophilization agent to make a nice kit file, and also solubilizers to ensure everything dissolves properly. This figure shows you just how the labeling works. So your pertechnetate comes out of the generator in a coordination state of 7. And so you have to change it to something more useful by adding this reduction agents. like status chloride to make it more useful, and for example system review use at coordination state 1. But what I want to show you with this figure is that there is ways where your technetium kit will have free pertechnetate, and that is when you have too little tin in the kit, when there is an error with the kit, when you have old eluate that contains technetium 99. So technetium-99 is the non-radioactive isotope of technetium-99M. And if you also have error to your kit, then this will lead to the technetium-99M coordination state, +7 coordination state. And of course, when you see that on your scan, you will have this classical pertechnetate scan influencing the correct scan that you wanted. Then on the other side, if you have water and things like that, you will also influence the reduction of tin, and you will have colloids, which goes to the liver. Then I will go through the quality control, where you have two systems normally, acetone and saline. Acetone shows you the amount of pertechnetate in your mixture and your colloids, and your technetium radiopharmaceutical, which is not associated to the organic phase, will get stuck at the bottom. And then for system 2, you will have pertechnetate and your radiopharmaceutical on top, and your colloids will then be on the bottom. You can then calculate every impurity and see this the actual yield of your radiopharmaceutical. Technetium radiopharmaceuticals biodistribute through the various phases. Sorry, mechanisms. So it includes compartmentalization, where you inject the radiopharmaceutical in a compartment which it doesn't cross. The passive diffusion that crosses some barriers that you can then see if these barriers still function or not. Filtration, like kidney agents, blockade, where you have small particles blocking certain capillaries so that you can see the distribution of blood flow. Chemisorption, like MDP going to the bone. Facilitated diffusion, such as is the case in the heart for sestamide. Active transport in the thyroid, secretion, receptor binding, and then of course also labeled cells that migrate. There's also a complete video on my YouTube channel on this following topic. We have newer technetium agents, and here I have an example of tetrutide, where we started to have technologies that allow us to radio label peptides, similar to those used in positron emission tomography for theranostic applications. These have more complex labeling and is also much more susceptible to all the problems that you can have during labeling, like the amount of tin in the mixture leading to unlabeled peptide. It's also sometimes more than one step, so it's not anymore the traditional kit vial where you just add technetium and you have a labeled compound, but it might require you to do multiple labeling steps. And then, of course, we have isolink, which is also allowing us to use by the isonitrile, the oxo chemistry to radio label peptides. And here I have an example of fibroblast activating protein, which we will cover a little bit later. I also have a more complex radio labeling system where you first prepare an intermediate, which is then used to label your final compound. And here we will go on to the present of radio pharmacy and currently the nuclear medicine landscape. So I grouped positron emission tomography under present because it is really the more novel radio pharmaceuticals are all currently utilizing positron emission tomography, especially in oncology, because it has just a higher resolution. Um, Naturally, this type of decay leads to the development of 511 keV gamma rays, which is in the opposite direction. And this allows us to already get a quite accurate three-dimensional location of the emission, and we no longer have to discard most of the rays through collimation, and we have more accurate detection and localization of the emissions, and of course very much higher resolution pictures, which is very useful to pick up smaller lesions in oncology. Often the question is how important in which application is this higher resolution. So for instance, if you are doing bone imaging and you image small metastases, it's really important to see all hundreds of metastases with a PET tracer, or is a single photon emission tomography or even planar imaging giving you all the clinical information that you need. As we said, it was also present in the past with the first scans done in 1935, where they had equipment that detect radioactivity and then um, make what make a dot on the paper where it is localized, and so they could do a PET imaging this way. Fluorine 18 was first produced in the the first cyclotron was installed in 1930s, and sodium fluoride was made in 1958. And here is a bone scan, as I discussed, with fluorine 18 in a healthy patient here. Really high resolution compared to SPECT or scintigraphy. And the first medical grade FDG was developed in the 1970s, which revolutionized metabolic imaging in multiple diseases and not only oncology, also inflammation, infection, and neurology. So a cyclotron produces many isotopes, but F18 is the most prominent, and hydrates are generated where the proton is accelerated using an electric field that change from time to time to positive and negative. This strips the foil. So there is a foil that strips the electrons, just leaving the proton. And this is then aimed at oxygen water that is used to produce fluorinating. FDG, as we said, is produced from this radionuclide, where you then also have a metabolic imaging base, basically. So every type of disease that uses more glucose as a source of energy can be imaged and visualized. And as glucose, FTG is taken up into the cell and metabolized by hexokinase, but then it gets stuck in the cell and not further used for energy. This naturally correlates with the wide distribution of FTG with high focalization in the brain and the heart. The production of FTG is pretty standard these days and straightforward, using nucleophilic substitution, where the fluorine 18 ion attacks the mannose triflate precursor. Cryptofix is used as a catalyst, and it enhanced the reactivity of the fluoride ion, and all of this takes place in anhydrous acetonitrile after the fluorine 18 has successfully been substituted. It is important to remove the protective parts of the molecule by a very basic or a very acidic media, and then you end up with the correct molecule FTG. There is many other fluorinating radiopharmaceuticals out there targeting many diseases like cell proliferation, which is also increased metabolism, like thymidine kinase 1, amino acid transports, also for tumors, F-dopa for dopamine synthesis, which is very well widely used for neuroendocrine tumors and Parkinson's disease, also targeting prostate cancer, breast cancer, and tumor hypoxia. All fluorine-18 production takes place with automated synthesis unit, but if you ever are in charge of building a facility, it's important to know automation can be used for almost all radiopharmaceutical procedures, perhaps with the exception of technetium-99m. And what automation does is it enhances the quality of the production, reduces the susceptibility to human error, increases the sterility aspects of your production, and it's just much easier, allowing you to have less personnel working with reactivity. So automation should be considered also for gallium and lutetium labeling, and is always 100% used in the case of the upscale production of fluorine-18 radiopharmaceuticals by cyclotron. Now we move into the more targeted approach on molecular level, and there is different vectors that is currently being used, ranging from full-size monoclonal antibodies with longer circulation times in the body, like 2 days or 4 days, fragments that are smaller that still tries to harness the specificity of antibodies without the problematic long circulation times, peptides most often used in theranostics, and then also small molecules more used for other diagnostics targeting other systems in non-oncology applications. We then move to the advent of gallium-68, and as you can see, gallium is used in a variety of peptide-based radiopharmaceuticals targeting diseases with this receptor expressions, and the well-known ones currently are PSMA, and then also the octreotides, octreotide, sorry, dotatok, dotatate, and dotatok. And then also we are getting a lot more application of fibroblast activating proteins, but there's also long-standing interesting targets like Pentixa for also bone targeting and HER2 receptors, glioblastomas, you name it. And there is many, many more in the pipeline. There's two ways to produce gallium-68, and I'm first discussing the generator-based ones, where you have the parent isotope with a 270-day half-life, germanium-68, loaded on multiple types of columns. Most often, the organic resins gives the less more stable systems, but you also have titanium oxide and tin oxide. You normally have to do some pre-purification process to make sure that you reduce the amount or the influence of metal contaminants, because gallium-68 is a metal and it's incorporated in the 3 plus charge state. Also very important not to use metal needles during your radio labeling process, and also adjust the pH in the end to around 4.5 or 3.5, ready for labeling. Most often, the limitation is the amount of radioactivity that you can elute from a generator, going to around 2 gigabecquerels, which can be at the start of your generator shelf life, around 3 patients. Then you have the cyclotron-produced gallium-68, where you can have either a solid target that can give you up to 194 gigabecquerel or a liquid target of 9 gigabecquerel. Same process, you have some a way of purifying your gallium-68 source through multiple separations, and then you adjust pH and you label. Then you also have just an example of how the biodistribution of gallium-68 looks like. And when it's not labeled, it is tend to associate with citrate in the blood and form blood pool imaging and distribute in a very characteristic pattern. So any unlabeled gallium in your mixture will give this as a background signal. You can also then elute the generator with the hydrochloric acid. And yeah, this can be done in automation or manually by using pushing or pulling through the hydrochloric acid through the column. Here I just have an example of a gallium kit as well. You have a lot less components, but it's important that this is also always happening in a metal-free environment. And we normally have some ascorbate buffer to protect against radiolysis and reactive oxidative species that can damage the targeting peptide. It's very important when you label with gallium to know that your method is not corresponding between the different generators. So if you have different sources of gallium, it comes out in a different acidic quality, and you might need different amounts of buffers to get it to the right pH. There is a lot more extensive quality control going into gallium-68 produced radiopharmaceuticals, looking at the appearance, the pH of the eluate. Normally, the presence of the parent isotope can be problematic, germanium-68 with long half-life, metal impurities, bacterial endotoxins, ITLC. Highly recommended to start doing radio HPLC, check for any residual solvents if used, and also the bubble test. There is also a series of ITLC strips. I'm just giving you some examples here, and these are also published in the European Pharmacopoeia. I want to caution you today about the presence of radiolysis, specifically also in gallium-68 radiopharmaceuticals, where a small impurity can have quite a big difference on the effective targeting of the peptide, where sometimes this radiolysis is not visible on ITLC, and that's why you should definitely invest in a radio HPLC if you are doing gallium-68 work. And then I want to mention to you the replacement of gallium-68 with a fluorine-18 metal, where fluorine-18 is reacted with aluminium chloride to form aluminium fluoride, which is then incorporated in the normal chelator system as with gallium-68 radiopharmaceuticals, and you get really high yields of 60 to 70%. So I guess in the future, the question will be, are we using fluorine-18 or gallium-68? And this depends from facility to facility. But if you are a new upcoming radiopharmaceutical institution that is being installed, it might be of better use to focus only on fluorine-18 and use this method to label peptides and new tracers with aluminium fluoride instead of putting in some separate gallium-68 infrastructure. Then, of course, currently there is the high movement of theranostics, and here I provide you the example of gallium PSMA, where after clinical translation happened in 2012 to 2019, PSMA was quickly labeled as well with lutetium and actinium to enhance the therapeutic use of PSMA. You see what you treat. So you have the same molecule where you diagnose or see the presence of the target into the patient, where after you exchange the diagnostic radionuclide with a more cytotoxic nuclide, and then you treat the same tumors that was visualized, and then you can use again the diagnostic counterpart for follow-up to see if the therapy is actually working and there is no disease progression. Lutathera was the first FDA-approved lutetium-based theranostic for gastroenteropancreatic neuroendocrine tumors. There is dotatate knock and talk targeting the neuroendocrine tumors, and it's very important to remember for therapy, dotatalk is less less recommended because it can have a higher radiation dose on the kidneys compared to dotanoc and tate, just because of the biodistribution and the affinity of the peptide for the kidneys. Further, it's not really clinically important which one of these you choose. Dotatate was, of course, the first one implemented in the clinic, but due to patenting constraints, knock and talk was developed and can be used equally. For diagnosis, there is no difference. Currently, there is also a lot of investigation being done about agonist versus antagonist in somatostatin receptors. So the agonist will always internalize and then irradiate the cells from inside, and antagonist will stay on the cell surface and disrupt the membrane. But then also it's important to remember that this is also a higher affinity situation where the antagonist has higher affinity for the receptors compared to agonists. And the suggestion is that you can then get less cytotoxicity, like kidney accumulation, maybe even a less dosage administered with a higher cytotoxic effect at the tumors. Pluvicto was then secondly registered by the FDA targeting PSMA, and it's the PSMA 617, but there is also now multiple other PSMA molecules like PSMA, INT, and so forth. We also have to always be aware of off-target distribution because this can increase side effects. So we also have the classical problem of PSMA targeting the salivary glands. And this is more irradiating the tumors. Just a note on the production method of lutetium-177. It's always important to talk about the carrier-added and non-carrier-added production of lutetium-177. What is the difference? Carrier-added is when natural lutetium-175 that is enriched is used to produce lutetium-176 and then 177. And this has a multiple mixture of lutetiums in there, of which the only one that we really want to use is lutetium-177. But since they are chemically the same, it's very difficult to separate. So you have a high amount of non-useful, non-radioactive lutetiums in your mixture, with your lutetium-177 being diluted in essence with non-radioactive lutetium. On the other hand, you can produce lutetium-177 by irradiating ytterbium-176 to make 177, which then decays and has a sort of a generator principle to produce lutetium-177 in a very pure form, not contaminated by any other cold lutetiums. And then this is, of course, giving you a high amount of all radioactive lutetium-177 at the tumor when incorporated into molecules, whereas the one that has the cold lutetiums have different lutetiums incorporated in the peptide and accumulating at the receptors. There's also a higher shelf life for lutetium non-carrier added, where you have higher specific activity, so it's 3000 gigabecquerel per milligrams compared to the lutetium carrier added, which is less than 1,000. And we calculated based on certificates of analysis that most often your lutetium non-carrier added has an extensive shelf life of even more than 10 days, where your lutetium carrier added can go out of specifications as early as 3 days post irradiation. When do we use carrier added? This is in more broadly targeted radiopharmaceuticals where there is a large area of targeting where the presence of cold lutetium will not influence, like in bone pain palliation, liver cancer, and treatment of inflammation. Carrier added, non-carrier added should be used in the targeting of receptors only. I left a lot of things out for current. I didn't want to go into, I mean, we can talk for hours about this big topic, nanotechnology, carbon-11, immunotherapy, infection. There's all these niche areas where radiopharmaceuticals are being used. But I was now going to go a little bit into the future of what are we busy with currently and more focused on. Of course, also really with a focus on therapy for oncology purposes. So, fibrous activating protein is a novel vector that focuses on the inflammatory part and the microenvironment of tumors and also inflammation, where you have these things called these cells called fibroblasts. That is a natural component of these malignant processes and gets activated and express certain receptors. So we are no longer targeting the tumor cell specifically, but rather the environment that feeds these tumors. Fibrous activating protein has been shown to be more appropriate than FDG in multiple types of cancer and also comparable to FDGs in others. So it might be in the future that FOPI could take a lot of the work from FDG being more appropriate and also maybe available in some centers where there is no cyclotron production. So this is really great news. Of course, FOPI cannot replace FDG in any way because FDG's application is much more widespread than just oncology, but it's nice that there is maybe a second option when FDG is not appropriate or not user friendly. And it's, of course, proven that in hematological cancers, lung cancer, and thyroid cancer, fibrous activating protein is still less informative than FDG. It has proven its worth in diagnostics, but we are still trying to optimize it for therapy because this tracer also have a high washout out of the tumor and there's multiple strategies to better design therapeutic counterparts. There is also a whole lecture on fibrous activating protein on this YouTube channel. I wanted today say that it's maybe time to rethink radiopharmacy because the future is a lot of growth and we have new molecules, new radionuclides procedures are getting more complex. And I think in the future we might want to look at the interaction between radiopharmacists and radiochemists and technologists in the radiopharmacy, adding some more personnel with more niche expertise and also just proposing some differences in the organization based on the level of complexity that you produce. Alpha emitters is currently gaining still a lot of investment just because of the shorter range, higher linear energy transfer, DNA damage. It has been shown to be effective in more effective in some patients, and there is the hopeful 8. It can be used, but in my opinion, there's only 4 that seems to be gaining traction at the moment due to availability. Radium-223 is also not maybe discussed in depth because it is more targeted on its own by a physiological process rather than getting incorporated into molecules targeting receptors. Astatine-211 is getting a network, but it's not so difficult to implement Astatine 211 with a relatively low investment in infrastructure needed compared to other production of alpha therapy. It can be produced by cyclotron. It's very important to get the energies that you use very good calibrated, not to produce longer lived contaminants. And then astatine 211 decay by one single alpha emission and has a therapeutic effect. We do have somehow a problem a little bit about chemical stability, but this is also being worked on. Astatine compounds form a little bit weaker halogen bonds than ionated counterparts because it is quite a heavy um, radionuclide, and there's also a little bit of a variation in the metabolism. Um, but this is being investigated with some clinical applications and some guidelines coming out in the production of alpha emitting MIBG called MIBG, incorporating astatine 211, and of course utilizing the natural accumulation of astatite into the thyroid using sodium astatite. Is also targeting going on with monoclonal antibodies for brain tumors and ovarian cancer. Actinium-225 is also one of the most widely used alpha emitters, and there is 3 production routes to make it, and we are still focusing on upscaling the production of actinium-225. On my YouTube channel, you will find a lot of discussions about actinium-225 as well. I worked with it myself extensively in the clinic. Most often, the 4M229 production with a generator-based system is the most published methods, but this is not sustainable for future upscaling, with the focus going more to the using of radium-226 in a smaller cyclotron system to produce actinium-225. There is some production at the moment reaching 63 gigabecquerel per year, but it's predicted to have a demand of more than double of that and a future production capacity of even more, hopefully resulting in some actinium-225 to spare. The quality control of actinium-225 is also a topic that I've extensively covered on my YouTube channel, but it's important to know that at the moment, most applied clinical procedures do not measure actinium-225 directly. The alpha emission is very difficult to measure, but instead measures the presence of daughters in this decay chain. And this results in a very complex quality control system where you do develop your TLC strips or your HPLC chromatographs, but only measure 1 or 2 hours after you have taken the samples and did the procedures so that the daughter radionuclide is present in equilibrium. If you do not measure later, you will lead to inaccurate purity, and maybe you will underestimate the purity of your compounds. Actinium-225 is hampered by lots of possible instabilities, of which weak-well energy is often discussed, where the decay of the alpha emission results in the complete removal of the actinium-225 the daughters from the system that targets it. So by the time you go to the bismuth-213 or the later polonium-213-209, these are completely not associated with the peptide that was used to target the radiopharmaceutical. But most importantly, the chemical properties of the daughters also change and leads to further instability. In my opinion, because of the long half-life of actinium-225, if you target it with peptides, this is not really that important. But what I worry about is, of course, the presence of radiolysis, where all this alpha emissions and reactive oxidative species in the mixture could damage the peptide. Even though the actinium-225 is still present in the chelator, this will no longer be effectively targeted. And that's why it's really important to do radio HPLC. And yeah, we will have a discussion on that later in the conference, but also on my YouTube channel. Clinical applications are growing every day. We have lots of applications currently being investigated in clinical trials, actinium-225 being the most applied clinical alpha emitter. Then lead-212 has a very promising future, also with the possibility of using it in generator-based systems, and lead-212, of course, emits both beta minus and alphas, so it is the best of both worlds, having the wider range of beta minus emission, but also the high toxicity of alpha emissions incorporated in one radionuclide. There is a lot of production going on worldwide. And it also has a therapeutic, a diagnostic counterpart, lead-203, that can be used for imaging. Numerous applications have been registered or reported, um, and this includes both small molecules and monoclonal antibodies and everything in between. Just a note on Auger emission, which also would have some future therapeutic applications, where Auger electrons has a higher linear energy transfer and damages the DNA directly. Very much multiple Auger emissions per radionuclide in the size or vicinity of one cell diameter. And this is where terbium radionuclides come in and provide a much more interesting therapeutic profile, with terbium-161 incorporating both Auger emission and beta minus emission. It's again a hybrid. But your terbium radioisotopes have multiple decay mechanisms like alpha emission, beta minus emission, SPECT, and PET. So it's a radioisotope family that incorporates all types of emissions, which makes it quite interesting. And here I just have an example of terbium-161 that is maybe in the future more strong than lutetium therapy due to this multiple mechanisms of decay for therapeutic, including Auger and beta minus. Rhenium-188 is also something that I see a future application for. Rhenium has the same chemistry compared to technetium, but it's less stable with more extreme reduction needed. And it's not really true that you can directly translate the same labeling methods, and chemists have been working really hard to figure out the chemistry. Oh, I was still recording. Chemist is really working hard to figure out the chemistry of rhenium. Here I have just an example of the current applications of Rhenium-188, and then there on top is me having my generator where I elute a Rhenium-188. You have ample production of rhenium available, where you have no limitation on the amount of rhenium, and it comes in a generator system that you can keep in your department for a month, oh sorry, for 6 months to a year, and every second day you can treat patients. And currently the applications include skin cancer therapy with a radioactive rhenium cream most often, but also liver cancer, as well as bisphosphonates. And of course, it's important that we will look at automation or cold kit methods, but then we also have newly investigated targets like octreotide, PSMA, and fibroblast activating protein. And here is just an example of the currently investigated rhenium PSMA that is looked as an alternative to lutetium PSMA. That's it for my presentation. Here I'm just mentioning the Onselium Foundation today at the conference as well. And yeah, if you have any questions, of course, you can pop it down into the comments. Thank you kindly for your attention.